Hey, Hammy here, back with part 2 of section 2.2, biochemical reactions. In this video here, we're going to take a look at little helpers for biochemical reactions to take place called enzymes. Something that helps speed up a reaction is called a catalyst. Okay? And there's all kinds of chemical catalysts, like in your car. You have a catalytic converter that speeds up the process of the gases coming off your engine, and it will change them into less harmful gases as they leave through your tailpipe, and it has to happen quickly. Okay? Proteins are biological catalysts, or enzymes, sorry. Enzymes are biological catalysts that speed up reactions. Enzymes are proteins, okay, one of the 4 biomolecules. They reduce the amount of activation energy. Remember in the last video we said that's a little kick in the butt, that little hump to get a reaction going. They reduce that amount of activation energy needed to get the reaction going. Okay, again, I said they're proteins, and over here you can kind of see example. Here's a kind of a quaternary shape of protein, might be several polypeptides in there, and here are our reactants right here that we're going to put together. Okay, there is one enzyme in the cells in your bodies for every chemical reaction that needs to take place. So you have a whole lot of enzymes in your body. And the enzymes are often named after the substrate that they break down and end in ase. For example, starch, though a fancy word for starch, is amylose. So guess what the enzyme is that breaks it down? Amylase. Okay? So oftentimes we take whatever is being broken down and we add ase to the end of it. Enzymes are used to regulate chemical pathways. Depending on which enzyme is present, it will take different paths and become different products, depending on what your body or your cells need. They can tear down materials, or they can build new materials, build new cells, new tissues in your body. They help release energy, and they also help transfer information in cells. How an enzyme works is called the Induced Fit Model. Back when I was in high school, we learned lock and key. So an enzyme had a specific shape. Okay, here's our enzyme down here, and we said it has a specific shape that fits the substrate that it's acting on. Now we kind of know it more as an induced fit. It's not so much like a perfect lock fits, or a perfect key shape fits an exact lock. It's more of an induced fit. This substrate you see kind of fits in there, and after the enzyme sort of hits the substrate, it will actually induce a shape change here. So it's kind of like the substrate hits the enzyme and then it kind of grabs hold of it. Okay? It grabs hold of it, it causes the reaction to happen, and if you look over here on the graph, how does it do that? It reduces the activation energy. So here's the normal reaction, the red line here. Okay? This hump here, remember we said, is E subscript A. Energy of activation. Look how much that lowers the activation energy with the enzyme. So that helps the chemical reactions to happen much, much faster. Some things that might take hours to occur will happen in days or split second, in a jiffy, we say. And it allows it to happen fast enough for us to have a faster metabolism, there's that word from the last video again, and helps us to essentially maintain life. Enzyme speed is regulated by 3 things. The 3 things, first of all, being temperature. Okay, remember the definition of temperature is the average kinetic energy of molecules, so how fast molecules are moving. Okay, if you increase the temperature, how do these enzymes work? Well, they're floating around in the cell, and remember the active sites need to hit and connect with a substrate. So if the molecules are moving faster, there's more of a chance that they'll run into each other and that the reaction will happen. So if you see on a typical graph here of enzyme activity, this is rate of reaction, this is an increasing temperature. So as the temperature goes up, our As our temperature goes up, our enzyme activity is going to go up. Usually the optimal rate is about 37 degrees Celsius. Why that kind of weird number? Because that tends to be body temperature. If it gets warmer than that, it causes what we say denaturation or denaturation. The enzyme will the disulfide bonds that's holding it in that tertiary structure will begin to break apart, and it loses its shape. Remember, form fits function. If it loses its form, loses its shape, the active site no longer binds to the substrate, and they don't work. So if it gets too hot, we say they get denatured. They don't work anymore. So we found that for most enzymes, they have this optimal rate of 37 degrees, which is about body temperature, 98.6 degrees Fahrenheit. The second thing that affects the speed of enzymes is the pH. How does that work? Well, different enzymes work at different pHs. We say they have different optimal pHs. Optimal is like where they work the best. Take these two enzymes down here, pepsin and trypsin, that break down proteins in our digestive system. Notice that pepsin works best at a pH of about 1 to 3 on the pH scale. Okay, 1 to 3 is a very low pH because pepsin is found in our stomach where you get hydrochloric acid, stomach acid. So this works the best where there is a low pH. Once it moves into the small intestines, okay, there it is buffered with the pancreas with bicarbonate ions that brings the pH up just above neutral to about 7 or 8. Okay, trypsin is found in the small intestine where protein digestion continues. Okay, but at a higher pH. Pepsin stops working once it gets to the small intestine. If trypsin were to get into the stomach, it wouldn't work. Again, outside of that pH range, they would start to lose their shape, and the active sites won't work. We say they would be denatured. Finally, the third thing that affects the speed of an enzyme is concentration. Okay, now concentration of two things. Number one, concentration of the enzyme that is doing the reaction, or number two, the substrates that are being broken down or built up. So obviously the more enzyme you have, the more reaction you will have. Or if you have just a couple enzymes, if you have more substrate, again, the chance of a collision inside the cell or wherever this is happening is much greater. So you'll see on a graph it looks a little bit different than temperature, but as you increase the concentration of an enzyme or a substrate, the rate of reaction over here, the rate of reaction goes faster, 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 and then it kind of levels off. You don't get any denaturing. You just hit what we call a maximum velocity. Okay, that enzyme is just working as fast as possible. Okay, as however fast that enzyme can cause a reaction, as soon as it lets go and turns around, okay, enzymes can be used over and over again. Every time it turns around, it's going to be having another reaction, another reaction, another reaction. Okay, it's like your car. If you mash the accelerator, it'll go faster, 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 faster. Eventually, you'll hit a maximum speed. Your car has not shut off. Your car is just traveling along at 100 miles an hour, and that's as fast as it could go. Very similar with an enzyme. Okay, again, enzymes are proteins. They're biological catalysts, and you need one for every reaction that's happening in your body. Okay, and they help speed up those reactions to make life possible.